Okay, so our bodies are full of tracking devices. We have chemoreceptors that are reading the chemical composition in our body on a regular basis. And we also have baroreceptors that are reading the pressure in our body. And then we also have thermoreceptors that are reading the temperature in our body. So the question is, where do we find each of these receptors? So in the case of the thermoreceptor, we can often find those temperature gauges, if you will, in various parts of the body. So, for example, we know that the normal body temperature is 98.7 degrees. And our body, our brain, is constantly disseminating the information that our body is feeding to it and then generating a response. But first, it has to collect the information from the body. So the thermoreceptors are the means of collecting the information. The thermoreceptors are buried in various places in the body, but the big ones that we can think of are those large blood vessels that are in our neck, namely the carotid arteries. They have thermoreceptors, specialized cells, that are able to pick up information about the ambient temperature outside and feed that information to the brain. The brain is then going to take that information and, for example, in this instance, realize that either you're too cold or you're too hot, and then the brain is going to generate a response to offset the shift from normal. And that process is called a negative feedback loop, which I think we've talked about before. So basically, whenever you skew off of normal even a little bit, your body is going to generate the opposite response of whatever that abnormal shift is in an effort to bring you back to normal again. So, in the instance of a patient or a person who's cold, the negative feedback loop there is to read the temperature in the bloodstream and send an impulse down the central nervous system, spinal cord, out to the peripheral nerves that shoot off from the spinal cord, and it's going to tell the body to start shivering. That nervous impulse will trigger shivering in the body. The shivering in the body is achieved by causing muscles to shiver, as we all know, and that is going to create heat that will then offset this drop in temperature. Now, if you're overly hot, the brain is going to send an impulse down the spinal cord through the peripheral nerves, and that will direct your body to start perspiring. So it's going to speak to the different glands, and in that instance, it will cause the glands to start perspiring, and it will also cause blood vessels in the skin to become larger to help disperse that heat. Okay, so it will cause the diameter of the blood vessels in the skin to vasodilate, and that will help disperse some heat, and then it will also trigger your sweat glands to perspire, which will cause the condensation and the cooling that will bring us back to a normal body temperature again. All right, so it's important to recall that both of these are considered negative feedback loops, both the sweating, the vasodilation in the blood vessels, and the shivering. Now, if we look over at another method we have of maintaining homeostasis, we can consider the chemoreceptor. So, chem means chemical, obviously, and the chemoreceptors that we're talking about in our body primarily today are going to be the chemoreceptors that measure not so much the level of oxygen, but actually the level of carbon dioxide in our bloodstream. And that's often a difficult concept for people to understand because it seems more sensible to them that it would read the level of oxygen, but in fact it's reading carbon dioxide. And it makes sense. Our body is constantly producing carbon dioxide as a waste product, so it's more important to track that. Now we have these various structures in the body. We've got the diaphragm there that we're noting, and in a resting position it's going to be in a relaxed position, and that diaphragm, the rate of contraction where it gets pulled down, as we can see with the dotted line, that rate of contraction is going to be dictated by the brain, and that's going to be based on the information that is fed to the brain from the chemoreceptors. So not only will the rate increase, but also how deeply a person breathes can be created, or dictated rather, by the level of carbon dioxide in the bloodstream. So we talked about how the diaphragm pulls down and it creates a vacuum because it increases the space from a small size of the lung to a larger size in the lung. That creates a vacuum, and air rushes in. Now, when we look at our level of carbon dioxide in the bloodstream, it's important to think of it as a thermostat. And we measure the amount of carbon dioxide in the bloodstream by millimeters of mercury. Because whenever you put something in a container that's full of something else, it's going to take up space and exert pressure. So carbon dioxide is in the bloodstream, it's being carried around by hemoglobin, and it's going to exert a certain amount of pressure. And the normal amount of pressure that's exerted is between 35 and 45 millimeters of mercury in the body. Now, if you guys have ever studied pH, you'll note that the pH can shift when the levels of carbon dioxide get too high. So if they get up to the 50, 55, 60, 65 area, that means our patient's pH is going to drop below 7.35. In other words, they're going to become acidotic. Carbon dioxide is an acid, and it can alter the amount of neurotransmissions in the body. We'll talk about that later when we get to neurology. So if the brain senses an increased level of carbon dioxide in the bloodstream, it's going to send an impulse through the spinal cord down to the diaphragm. That impulse is going to direct the diaphragm to contract. And whenever something contracts, that requires work. Okay, that's an expenditure of energy. So the brain is going to direct the diaphragm to drop down, and if the carbon dioxide levels are pretty high for some reason, it's going to direct the diaphragm to do it at a faster rate. So think of an instance where your carbon dioxide levels might be high. If you've just finished running a race, all that muscle has generated carbon dioxide. Now, let's say your carbon dioxide levels are within normal limits, right there at 40 millimeters of mercury. Then your diaphragm is going to direct, or your brain is going to direct your diaphragm just to contract at roughly 12 to 20 times a minute. That's your normal respiratory rate. Okay? All right, so let's move on to baroreceptors. Now, baroreceptors, they sense the amount of pressure in the body. Now, pressure can be important for a number of reasons. Okay? Pressure will help dictate when to stop breathing, for example, because in your lungs you have these pressure receptors in your lungs, or stretch receptors, and when your lungs have inflated to a certain point, they'll trigger the brain, feed that to the brain, and the brain will say, okay, stop inhaling. All right? But there are other areas where baroreceptors are at work, and primarily these baroreceptors, or receptors that sense pressure, can also be found in the aortic arch. Okay, so the aortic arch, what it's sensing there is the amount of volume of blood in the aorta, right? So if these baroreceptors sense a loss of pressure, meaning that the volume of the blood vessel in the aorta just isn't as full as it should be, it will send that information up to the brainstem, and the brainstem can elicit a response as well. So if the pressure were low because perhaps someone got injured, they had a nail stuck in their arm, okay, and they're bleeding profusely from this horrible, horrible injury. Because blood is oozing or rapidly leaving the blood vessel, the pressure inside the blood vessel is going to drop a little bit. Now that information is going to get fed to the brain that there's a loss of pressure in the entire vascular system. That information will be fed to the brain, excuse me, and the brain will then elicit a response as well. So one of the responses might be to direct the flow of blood to essentially make the container smaller again. Because if the container is full, then the fluid inside it is going to be able to travel and circulate better than if it's only partly full. So what the body will do is the brain will direct the blood vessels to vasoconstrict. Okay? So that means to get smaller. And in directing the body to do that, collectively throughout the body, now we've taken a container that is missing some fluid because this person was careless doing their work or whatever happened. It's vasoconstricted the blood vessels, thereby making the container smaller, so now the container is completely full again, so that even though there's less fluid in there, that's okay, the body will make more blood if it decides that it needs it, but until such time that it takes to do that, the blood vessels are smaller, the container is full again, and now the blood can circulate at optimum ability, where it might be at suboptimum when it's partly full. Even if it's mostly full, it's still going to not operate at the same optimum ability. All right, so we know we've got the baroreceptors, we have our thermoreceptors, we have our chemoreceptors, and all of them are meant to help us maintain homeostasis, because if anything shifts on the exterior of our body or on the interior of our body, that can throw us out of balance. So that information, whether it's a change in the interior of the body, like the loss of blood, or the exterior, like the cold, that information gets fed to the brain. The brain will then send impulses to various parts of the body to deal with whatever the issue is, whatever the change is, and then that is how you maintain homeostasis. So let's move on to some other topics. These can be a little bit tough sometimes for some people, but they're important. So when we're exchanging carbon dioxide and oxygen in the bloodstream, it's constantly taking place every time we inhale and exhale. The question is, why does it happen? Well, one of the principles is the concentration gradient. Another is the semi-permeable membrane, where there are openings in the membrane. And both of these will help to define or delineate how the exchange of carbon dioxide and oxygen will work. So in the semi-permeable membrane, also known as the membrane with holes, it's a good way to think of it, what it's going to do is permit some things past it, and it's going to bar other things from traveling to the other side of the membrane. And basically what it boils down to is how big are the molecules or the compounds on one side of the membrane, and how small are they, and then how big and how small are the holes in the cell wall. So cell walls have a semi-permeable membrane. Blood vessels have a semi-permeable membrane. And they all are selective, meaning they will only let certain size things in and certain size things out. And that will help to maintain homeostasis as well. So if we look at, for example, the semi-permeable membrane how it relates to the concentration gradient, we'll find that not only will the semipermeable membrane let things in and out based on their size, but also what is going to travel from one side to the other will be determined by the concentration gradient. How much of a certain molecule is on one side of that membrane compared to the other. So when we're talking about the movement of particles, it's going to be dictated by both of these concepts. Now, when we talk about osmosis, we're talking about the movement of water molecules. And we'll draw them like these little Mickey Mouse ears, okay? Because you have two molecules of oxygen with one of hydrogen, and that's typically how it's written out. So the molecules of water are going to shift back and forth in an effort to achieve the same concentration of water versus particles on both sides of the membrane. Okay, and that's going to be largely dictated by the concentration of water on one side, as well as the concentration of other kinds of particles on the other side. And those other particles could be proteins, they could be fat molecules, and the proteins can also simply be structures in the cell. So the movement of water through the semipermeable membrane is osmosis, and it's going to be controlled by the concentration of water as well as the concentration of particles on either side of the membrane. Now, if we consider that diffusion is a similar concept, then we can go from there and understand that the concept of concentration applies there as well. So to explain the concentration gradient, let's say that this square thing that I'm trying to draw up here is actually a dish, a rectangular dish. But one side of the dish is deeper than the other side of the dish. And if you were to pour water in it, where would it go? It would run downhill to the deeper end, right? And as we continue to do that, then the water would be deeper on one end than the other. It would be more concentrated on one end of that bowl than the other end of that bowl. So there's a high concentration in one end and a low concentration in the other. And that's basically the way everything else is going to move, is that it's going to move from a downhill perspective. It's going to move from the high point down to the low point. So the bowl in this case is, the high point of the bowl is the shallow end, the low point is the deeper end. And the same is true when we're talking about the concentration gradient of particles. If there's a whole bunch of particles on one side of the membrane and far fewer on the other side of the membrane, they're going to seek to equalize. Okay, so when particles of water are different on either side, let's say on one side there's 10 particles of water and on the other side there's 3 particles of water, and on each side there's 2 particles of protein, then the water is going to try to shift over to the other side so it has the same number of particles of water compared to the number of particles of protein. Now diffusion is actually the movement of particles through a semipermeable membrane. Now, it's only going to happen if the particles are small enough to fit through the holes. So it's selective in that regard, and a lot of semipermeable membranes are selective, meaning they're just the right size to let certain particles through, but just too small to let other particles through. Nonetheless, particles are also going to follow the concentration gradient when they attempt to travel through a semipermeable membrane. So if we have a number of particles over here in purple on the one side, and there's an unequal number on the other side, if those particles are able to cross through that membrane, they will try to do that until they equalize on the other side. So if you had carbon dioxide particles on one side of the membrane and oxygen on the other, the concentration gradient says that since they're high on one side and low on the other, they'll try to switch. Now this is also true in our bodies for Sodium, for example, we have high concentrations, we have concentrations of sodium in the body, and the movement of sodium and potassium in particular, but mostly sodium, is going to help dictate a number of properties in the body. So when sodium travels, it can make it through this semi-permeable membrane. It's a small little ion. And then water will attempt to travel as well. And as they both try to travel, they're both trying to equalize on either side of the cell wall or the semi-permeable membrane. And they will attempt to equalize so that you end up with the same number of molecules on either side. If there's a disparity, meaning something's unequal, then it's probably because they just haven't finished the process yet, if you will. However, there are some processes in the body that will bank on keeping that unequalness, on either side of the semi-permeable membrane, because when you equalize, you also lose energy. Okay? So anytime something equalizes, we lose energy. So there are certain functions in the body where keeping it slightly off kilter means that there's the potential for energy there. And we'll talk about that later as well. Now let's say we've got particles in the bloodstream that we've drawn up here, and we can see that they're obviously large. And it might be a good idea to think of these particles as sugar. Okay, sugar is something that we utilize, we have to have. It's our major source of fuel in our body, and our body will take those molecules of sugar, break them down, turn them into cell food, and cellular energy is produced called ATP. Now, in osmotic pressure, what's interesting here is because these molecules of sugar are too large to pass through the membrane, they're going to sit on one side, unless something can help them travel to the other side, which we'll talk about later. But if they can't travel to the other side for any reason whatsoever, then they're going to sit on that one side, and they're going to pull water to them. That ability to pull water to particles is called osmotic pressure. And usually when we think of pressure, we think of something pushing. But with osmotic pressure, it's important to think of it rather as something pulling. These particles can pull molecules of water to them and will often be in opposition to the concentration gradient. So you've got the concentration gradient that's telling you that everything is going to move from a concentration of high to where it's a lower concentration. But if you have particles trapped essentially on one side of the semi-permeable membrane, they are going to have an effect on the water molecules on the other side and will pull the water molecules to them. And that's called osmotic pressure. So I want you to think of what those particles might be. They could be proteins, structures in the cell, molecules of sugar, anything that is so large it can't pass the other side.